So we're happy today to hear Carl Philipp Heisenberg from IST Austria, and he will tell us about tissue rigidity transitions. Please, Carl Philipp.
Okay, thank you very much. I see a largely empty lecture hall. I see, yeah, a few people in there. So what I would like to speak about is tissue rigidity transitions in early zebrafish development. And the work I'm speaking about is done by two postdocs. One postdoc is Nicoletta Petridou. She was in my lab, and she is now a group leader at EMBL. And the other one is Bernardo Minas Motra, and he was a member of the Hanesso group at IST Austria. Before I tell you about what we have done on rigidity transition in zebrafish gastrulation, I would like to give you a little bit of background on what we in the lab are generally interested in and what we have done over the last perhaps more than 20 years now. We have sort of a long-standing interest in understanding how forces are being generated in the early zebrafish embryo. And the one example I put in here is an early oocyte in which the yolk granules and ooplasm are segregating. And we are looking at force generation predominantly in the context of actomyosin contraction and flows. And what you can see here is an example where actomyosin flows, which are directed from the vegetal pole up to the animal pole, lead to a complete segregation of ooplasm and yolk granules by taking along the ooplasm more than the yolk granules and thereby triggering a segregation. So this is one example where we have looked at actomyosin flows and force generation. Another example, another area we are quite interested in is force transmission and balance within the embryo. And the example I put in here is a pretty old example where we're looking at contact formation. We were asking how big, what determines the size of a contact between two cells which are taking out from an early zebrafish embryo. And we ended up with a description where we define the size of the contact by the ratio of interfacial tensions. And one interfacial tension is the cell cortical tension at the cell medium interface, and another interfacial tension is the cell-cell interfacial tension, which is determined by adhesion tension and by cortical tension. And what we ended up here is that the main factor determining the size of the contact is the ability of these cells to decrease their cell-cell interfacial tension, and they do that predominantly by reducing the actomyosin cortex or modifying the actomyosin cortex at the cell-cell contact area. And by doing so, cell interfacial tension is being reduced, and thereby the ratio of cell interfacial tension between the cell medium and the cell interface is changed, and that determines then the size of the contact. And this is again different between different cell types and can determine then differences in tissue surface tension, for example. Another more recent area we are interested in is mechanosensation, and in a very broad sense of the word, mechanosensation in any sort of interaction between mechanical and chemical signals. One example I put in here is a cell again in the early zebrafish embryo. Now we're looking at the surface epithelium of a zebrafish embryo, which is called the enveloping layer. It's a simple squamous epithelial cell layer. And this is one cell which is about to divide within the plane. And the mitotic spindle is indicated here by this white bar here. And we were asking how the distribution of tension within the plane of this tissue determines the orientation of the cell division, the orientation of the mitotic spindle. And the experiment, and I just put in this one example we did here, is we induced an ectopic tension axis by plating cells here and here. So you generate a lot of tension between these two extrusion sites. And what you find is that the mitotic spindle now aligns along this newly induced ectopic tension axis within the plane of this epithelial cell layer. That's sort of an indication that this cell is mechanosensing, and it senses the main axis of tension within the plane of the tissue and orients the mitotic spindle along this new tension axis. Now, these are just three examples of what we very generally are interested in. What I would like to speak about is a very simple process, superficially very simple process, where we're looking at early tissue spreading. Essentially, what we're looking at here is again a zebrafish embryo, and it's roughly 4 hours old, and it consists out of 1,000 cells, which is forming the blastoderm, and a very large cell on which this blastoderm is sitting, which is called the yolk cell. And now what is happening in the first hours of morphogenesis is that the blastoderm begins to spread around the yolk cell. it appears as if the yolk moves into the blastoderm, but in fact, the blastoderm is spreading around the yolk cell. This process is called doming because it looks like a dome. But it's essentially just describing a spreading behavior of this tissue around the yolk cell here. It looks superficially quite simple, but we really wanted to understand it mechanistically in detail. And what we did is and I just introduce here the different cell types we are looking at in this process. It's quite simple. We have the yolk cell here, very big cell. One big cell. It has a thin layer of cytoplasm on its surface, which contains nuclei and various cytoskeletal structures, mitochondria, ER, you name it. And then on top of the yolk cell sits the blastoderm, and on top of the blastoderm, the surface of the blastoderm, these are deep cells, mesenchymal cells, and on the surface of the blastoderm, you have a thin layer of squamous epithelial cells, which are called the enveloping layer cells. And essentially, the interaction between these three cell types, deep cells, enveloping layer cells, and the yolk cell, determine this spreading behavior here. And this is just what I introduced to you now. And a number of years ago, a postdoc in the lab, Hitoshi Morita, he, together with the group of Guillaume Salbreux, we identified the main force-generating processes by which the blastoderm spreads around the yolk cell. And in one process, we postulated that the surface epithelial cells, these EVL cells, are undergoing active expansion. So they're flattening out within the plane. And by undergoing active expansion, they are lowering the surface tension of this tissue. And by lowering the surface tension, this induces the spreading behavior of the tissue over the yolk cell. The other force-generating process we postulated is active intercalation of deep cells along the radial axis of the tissue, essentially cells moving along the radial axis here and undergoing intercalation, thereby thinning the tissue along this dimension here and then extending it along the other two dimensions. So this active radial intercalation should be postulated as triggering an active anisotropic tension stress within the tissue. And this then leads to and contributes together with the expansion, together with the reduction surface tension, the spreading of the tissue of the yolk cell. So this was approximately 2017 when we finished this work, and there was another postdoc joining after that, Nicoletta Petrino. And she got interested in understanding the contribution of this active radial intercalation and tissue spreading and spreading of the blastoderm. And the first thing she did is she looked if these cells are indeed undergoing active radial intercalation on a cellular level. And what she noted is that there is actually right at the onset of doming, when the tissue begins to spread, there was very little indication that these cells are actually actively undergoing radial intercalation. There was no protrusion which pointed along the radial axis. These cells appeared much more passive than we originally thought they are. So that sort of indicated and posed a problem that this proposed mechanism of anisotropic stress generation by active radial cell intercalation is presumably not at work at early stages of doming. So we had to look closer into it and see how we can get doming in the absence of this process. There was one more experiment which Hitoshi still did before he left the lab, and he took the embryo and he depleted a large part of the internal cells, these deep cells. So he essentially generated an embryo which consists of surface cells, a very big yolk cell, and a few deep cells left in there. He did this experiment to reduce the amount of radial intercalation and force generation by the deep cells. And then he was asking if the embryo still can undergo doming. And what he found, and that was quite surprising, is that the embryo can actually nicely dome in the absence of most of the deep cells. He just replaced these deep cells with buffer in this case. Again, supporting the view, which Nicoletta also sort of acquired early on in her studies, is that this radial deep cell intercalation seems to be not a decisive factor of the early spreading of the tissue over the yolk cell. Now, the question we then ask is what triggers doming in embryos which have minimal active radial deep cell intercalation? And what Nicoletta then postulated is that a change in tissue material properties might be involved in this process. And the reason why she proposed this comes from a number of simulations we did together again with Guillaume Salbreux, where we looked at the embryo here in a supracellular manner here, where we are just looking at the blastoderm as one tissue, and the yolk cell. And what we are doing is we are assigning now different viscosities to the blastoderm tissue. And we are asking to what degree the blastoderm would spread over the yolk cell, here essentially driven by the balance of forces at the contact line or contact point here, and ask if we change the viscosity of the tissue, would that have an effect on the doming movement of the blastoderm? In this simulation, and this is quite important, we assume that the only force generating process would be a reduction in surface tension, but we are not putting in any anisotropic stress generation within the blastoderm. And now what happened is, if we assume that the viscosity is high and uniform within the blastoderm, and we only reduce the surface tension, then this blastoderm in the simulations is not spreading very far. Here we're looking at the relative height of York to the blastoderm here. And in the simulations, there's essentially no spreading visible. This is the experimental observation where the blastoderm begins to spread. In another scenario, we reduced the viscosity of the tissue and assumed here that the tissue has a low uniform viscosity. And then we asked if the tissue can spread only by reduction of surface tension. And again, there was very little spreading, a little bit more than in case of high uniform spreading. But this, again, didn't really match our experimental observations. Then, and this is Sylvia Grigolon in Guillaume Salbru's group, she did a number of different combinations, and what she ended up is one combination where things looked a little better. And in this case, she assumed that viscosity is low in the center of the blastoderm, but remains high in the margin of the blastoderm. And in such case, she could get to a behavior in her simulations which matched quite closely the experimental observations. Not perfectly, but she was much closer to the experimental observations. So this was a good prediction of what could actually happen in the embryo to trigger blastoderm spreading, even if radial deep cell intercalations are not working. So what Nicoletta then did is she used a micropipette aspiration assay to determine the viscosity of the deep cell tissue, essentially by aspirating deep cells into the pipette. And she did it at different stages before the onset of doming and after the onset of doming. So these two stages are before the onset of doming, and this is after. And this is right when doming is setting in, when the tissue begins to spread. And what she found, to her initial surprise, is that indeed the tissue in the center of the blastoderm is fluidizing right at the onset of doming, while in the margin of the tissue is not fluidizing. It remains, the viscosity remains essentially unchanged. And then at later stages, the viscosity goes back in the center, and you're ending up again with a state of viscosity which is very similar to the state before the doming has begun. Nicoletta could also take out tissue from the center of the blastoderm, took it into culture, and even this tissue autonomously underwent fluidization, as you can see here, right at the point when the blastoderm in the center would fluidize. Okay, so this sort of fitted with the idea that a fluidization of the tissue in the center of the blastoderm could be the trigger of blastoderm spreading in the absence of radial deep cell intercalation, only driven by surface cell expansion. Now, to test that functionally, what Nicoletta did is then a number of transplantation assays. In one case, this is just a control transplantation. She takes deep cells from the center and puts it into a host embryo, same stage, nothing should happen. Then she took cells from the margin, which are not fluidizing. This tissue in the margin is not fluidizing, puts it into the center of a host embryo where the center would normally fluidize, but now these cells are not fluidizing. And she did a third condition in which she take an embryo at an earlier stage, which is not yet fluidizing in its center, and puts these early cells into a late embryo, which is about to fluidize in the center. In these two conditions, she assumed she has now a mosaic tissue where part of the tissue is fluidizing in the center and part of the tissue, the transplanted tissue in particular, is not fluidizing. And then she's asking if she inhibits now locally fluidization in the embryo, would that have an effect on the doming movement of the blastoderm? That's what the original hypothesis was. Now, these are three examples of these transplantations. This is a control transplantation. Heterotypic means margin into the center, should not fluidize the green cells. And heterochronic means early tissue into the center. Again, these cells should not fluidize. And in the two experimental conditions, what she found is that the tissue indeed right, that the doming of the tissue close to the transplantation site was transiently but clearly inhibited. This sort of indicated that indeed fluidization of the within the center of the blastoderm is required for proper doming movements of the blastoderm, for proper spreading of the blastoderm. This is just a quantification of these effects. She did one important control experiment where she is aspirating the cells which she had transplanted, And indeed, she finds that these transplanted cells, either coming from the margin or from an early embryo, are not fluidizing while in a control transplantation, the tissue fluidizes. Now, this was sort of some functional evidence that fluidization within the center of the blastoderm is required for proper spreading of the blastoderm. Now, the question, of course, she asked is, why is the tissue fluidizing in the center? And the obvious thing to look at is the ability of cells to form contacts and changes in contact between cells during the point of tissue fluidization, when the tissue is changing its fluidity or viscosity. Now, in the center at high stage, when the tissue still has a high viscosity, the deep cells have pretty much continuous contacts with neighboring cells. And if the tissue is fluidizing right at the onset, when the viscosity goes down, these cells, and this is perhaps not too surprising, lose their contacts. In the margin, this is not happening. The margin, you have these cells are in pretty much uniform contact with neighboring cells, and this is not changing at the onset of blastoderm spreading. Now, if you look in a slightly more quantitative manner at it, you can look at contact time, for instance, or contact length. And both contact time and contact length go down in the center. You can see it here right at the point when doming begins. And this is also visible here with contact length, while in the margin, you don't see these effects. So essentially what that only means here is that cells are losing their contacts in the center of the blastoderm right at the point when the blastoderm is fluidizing. So the question then Nicoletta asked is, why and how is cell-cell contact loss restricted to the central blastoderm? Why is it only happening in the center? And why and how are cells losing their contacts in the blastoderm center? So what is the mechanism of contact loss right at the stage when the tissue is fluidizing? Now to the first question, why and how is cell-cell contact loss restricted to the central blastoderm? What Nicoletta then focused on is a signaling pathway, which is called the Wnt non-canonical or Wnt planar cell polarity signaling pathway. Had originally been identified in Drosophila to determine planar polarity within epithelial tissues, and subsequently also in vertebrates to be important for cell migration and cell adhesion. And what Nicoletta noted, and we have noted before as well, is that the Wnt PCP pathway, the signaling pathway, is switched on right at the point of blastoderm doming movements within the margin and only in marginal regions of the blastoderm, but not within the center. And the pathway looks like that. This is one of these typical schematic diagrams where you have a ligand acting on physical receptors, and then it goes down to cytoskeletal polarization and the activation of target genes. Now, what Nicoletta then wanted to know is if Wnt PCP signaling is required in the margin for inhibiting, for essentially allowing this tissue not to fluidize, for making sure that the tissue is not fluidizing, while in the center where Wnt PCP signaling is not on, the tissue would fluidize. She addressed the function of the Wnt PCP pathway by doing a number of transplantation assays. And again, what she did is she took tissue, and this is a control experiment, from the margin of a wild-type embryo, which is not fluidizing, and puts it into the middle of a host embryo. Again, in this case, she should see a reduction in doming movement, and that's exactly what she found here, because this tissue is not fluidizing. Then she took tissue from the margin of a Wnt signaling defective embryo, a mutant embryo for the Wnt PCP pathway. And she took now tissue from the margin of this embryo and puts it into the center of an host embryo. Now, what she noted is that this tissue, which she took from the margin of a mutant embryo, is actually fluidizing. So it's ectopically fluidizing, even that it comes from the margin, it's ectopically fluidizing. And once it is ectopically fluidizing, it's not inhibiting doming movements anymore. So it behaves like a central tissue here. And she did one level of complexity further up, is taking the mutant tissue, which is ectopically fluidizing, coming from a mutant, and then trying to rescue it by activating a downstream effector gene, in which case is Rho A, a constitutive active form of Rho A. Now this transplanted tissue now should behave like a wild-type tissue because it is mutant but rescued. And in this case, again, this tissue is not fluidizing. As you can see over here, the viscosity remains high, and doming is partially suppressed again in these embryos. So this and a number of other experiments indicated that indeed the activation of Wnt PCP signaling within the margin of the blastoderm makes sure that this region of the blastoderm is not fluidizing, while fluidization is then restricted to the blastoderm center. Now, the key question then to answer really was, why do cells lose contacts in the blastoderm center? And the reason for that comes by looking at a slightly earlier process happening in development of the zebrafish embryo. What the embryo does is it undergoes to a number of synchronous or metasynchronous cell cleavages, subdividing the volume of the blastoderm into smaller parts. And during the last round of cleavages, which are metasynchronous, or where the cells divide roughly at the same time, you can see that these cells are undergoing the last round of metasynchronous cleavages, and while they're undergoing mitotic rounding, they're losing contacts in the center, but they're not doing so on the margin. So what we speculated is that perhaps the fact that cells are going into mitotic rounding and then undergo cleavages leads to a transient loss of contacts, which is rebuilt quite efficiently in the margin, but which cannot be reestablished in the center of the blastoderm. So essentially, that cells, the fact that cells are undergoing their last round of synchronous cleavages leads to contact loss in the center, but not in the margin. Now, the way Nicoletta addressed it is again through these transplantation assays, and the logic here is a slightly different one. What she did now is she took tissue from an embryo which is not undergoing their last round of cell cleavages anymore by inhibiting S phase. This is using a mix of S phase inhibitors, and in this case, the transplanted tissue is not undergoing the last round of cleavages anymore, and this tissue should not, from the donor embryo, should not fluidize because it's not undergoing the last round of division. And indeed, this tissue is not fluidizing and it's partially suppressing doming movements. And she did another experiment in which she expressed a dominant negative form of esrin, an ERM protein, which in cell culture had been shown to be required for cells during cleavages or division to undergo mitotic rounding. So if mitotic rounding is important for contact loss, then if you express a dominant negative form of this esrin, this tissue should not, the cells should not undergo mitotic rounding, and consequently, they should not fluidize because they're not losing their contacts. And then this tissue is ectopically not fluidizing and should inhibit doming movement. And that exactly was, again, what happened. So she had two conditions here where typically the cells which she transplanted should fluidize because they come from the center of a donor embryo, but they are not fluidizing because in one case, they are not undergoing the last round of synchronous cell cleavages. And the other case, they cannot properly undergo mitotic rounding anymore. And that sort of provided experimental evidence for the assumption that the last round of cleavages and associated mitotic rounding is important for the central tissue to lose contacts and fluidize. Okay, this is published a number of years ago already, and what we ended up here is a scenario where we speculated that the last round of mitotic cleavages leads to a transient fluidization of the central tissue, but not the marginal tissue. And the reason that it's only in the center and not in the margin is that you have in the margin Wnt PCP signaling, which elevates or promotes cell-cell contact formation and allows this tissue to reform their contacts much faster than the case within the center of the blastoderm. Thus, only the center is fluidizing and not the margin. Now, this was when Nicoletta, when we started to collaborate actually with Eduard Tanesso and his group here at IST Austria. And Eduard got quite interested when he saw our data on the tissue fluidization. And being a physicist, he thought that what we are seeing here, this very rapid and abrupt and drastic fluidization of the tissue, could potentially have some features of a phase transition, where you go from a rigid to a fluid tissue in a very, very short time. And essentially what we wanted to know in this collaboration is then if what we are observing here in the embryo in the center of the blastoderm, this rapid and drastic fluidization of the tissue, might have features or might display features which are consistent with the idea that the tissue might go from a rigid to a fluid state. The control parameter would be, in this case, connectivity of cells, and the order parameter would be the rigidity of the tissue. And that there's a critical point in the connectivity of the tissue below which the tissue would be fluid and above which the tissue would be rigid. This was obviously a very sort of simplified and hypothesis-driven assumption, and we had at this point no evidence for that whatsoever. Now, the first thing what we wanted to do is to see if there could be indeed a phase transition or rigidity transition being involved is looking at the connectivity of cells within the blastoderm center. From now on, I'm speaking only about the blastoderm center. And the way we determine connectivity is now in 2 dimensions. One could do it, we have done it also in 3 dimensions, but for the sake of simplicity, what we looked here is a two-dimensional cross-section of the blastoderm center. And what we did is we connected cells to their neighbor in case they have a contact, meaning that the cell is directly adjacent to another cell and there's no interstitial fluid in between the cell. Then we are connecting the cell via a link to the neighboring cells. In case where there's interstitial fluid between these cells, like here, there's no node and there's no link between these nodes. So you're essentially arriving at these connectivity networks, these two-dimensional connectivity networks. And you can do it at different stages before fluidization, right when fluidization is happening, and after fluidization when the tissue is sort of rigidifying again. And then you can just calculate on these networks, you can calculate the average connectivity. And this is very much in line with what Nicoletta did before on contact size and duration, that there seems to be a slow and nearly linear decrease in the connectivity up to the point where the tissue is fluidizing, and then the connectivity goes up again when the tissue is rigidifying. But if you look at the viscosity as a function of connectivity, and this is quite important, we found that there is a critical level of connectivity from where on the viscosity of the tissue very sharply rises, and below which the viscosity of the tissue is very, very low. So that is sort of in line that connectivity might be a control parameter which determines the order parameter, which in this case would be viscosity. So there's, again, no evidence that this is indeed a phase transition, but it points in the direction that this could be resembling a phase transition to a certain degree. Now, this is when Bernard was joining, and he thought we can perhaps look at this phenomenon in the framework of rigidity percolation. And essentially, this is just an artificial network of nodes connected by links. And what Bernard did then in these networks, where one node can have a maximum of 6 connections to neighboring cells, he was asking if he's taking out random links in this network, how would that affect the rigidity of this two-dimensional network of interconnected nodes here? Essentially, the resistance against deformation. And he subdivided or color-coded here these networks where he marks floppy areas as areas which can deform quite easily, rigid areas as areas which cannot deform when you compress this tissue here, and the giant cluster, which would be the biggest rigid area within this network here. And what he then plotted is how big the biggest cluster, this giant cluster, would be as a function of the normalized connectivity in such artificial connectivity networks, two-dimensional connectivity networks. And this is all predicted, and this is not really new, is if you reach a certain connectivity value, then the size of the biggest rigid cluster, the giant cluster, rapidly increases. And if you go below, then the size becomes very, very small. So this is absolutely not new, but he just repeated what many have tried before on these networks. But the interesting part then comes when we constructed now these two-dimensional connectivity networks for different stages before fluidization, when fluidization is happening, and after fluidization when the tissue becomes rigid again. Now these are now experimental networks where we are looking right at the tissue within the embryo. And what then Nicoletta and Bernard did is they defined the size of the largest cluster, the giant cluster size, at these different stages before fluidization, when fluidization is kicking in, and once the tissue becomes rigid again. And from this analysis, and this was very encouraging, is that if you look at the normalized connectivity in these networks versus the giant cluster size, you see again, and this is perhaps not too surprising because it's just following what the theory would predict here, is that there is a critical connectivity value from which on the giant cluster becomes very big, above which the giant cluster becomes very big, and below which the giant cluster is very, very small. But what is more interesting is then looking at the viscosity as a function of giant cluster size, or more importantly here, viscosity as a function of the normalized connectivity. Then you see that there's indeed a critical level of connectivity in this network, which agrees exactly with what has been proposed here, which fits with a decrease or increase, rapid or drastic decrease or increase in tissue viscosity. You can also plot that in time. You see that the tissue goes in fluidizes, and then it sort of re-solidifies over time. So it goes twice over this critical point here. Okay, that's exactly when Nicoletta then wanted to see if what they found now as a function here of developmental time is actually she can experimentally manipulate. And the idea is now, if this critical connectivity, this critical point of connectivity determining viscosity would be a meaningful and general principle, then this should be independent of any experimental manipulation you're doing to the tissue. It should be always visible at the same point. And she did one experiment, which is the obvious experiment. She knocked down E-cadherin, which is the prime adhesion molecule, and thereby she reduced connectivity between cells within the embryo. And she did it before, right at fluidization and after fluidization. Then they constructed these 2D connectivity maps, and they asked what would be the size of the biggest cluster in these different conditions. Importantly, in all these conditions, the tissue has actually a very, very low viscosity. And what she found, and which agreed with that, is that the size of the giant cluster is always very, very small. In some cases, it can go above the critical value of connectivity, and then it becomes big again, but generally it stays below. Now, what then Nicoletta did, she took all sorts of experimental conditions. E-cadherin knockdown, she looked at, she interfered with myosin activity, she took tissues from different parts of the blastoderm, marginal blastoderm, mutant background, mesoderm-induced cells, essentially everything she could do with the tissue. And then she plotted connectivity as a function of cluster size, and even more importantly, connectivity, viscosity as a function of connectivity, and here giant cluster size as a function of connectivity. And she found always this transitory behavior where when she goes below a certain value of connectivity, the critical point, the tissue is essentially fluid, and if she goes above, the tissue is rigid. So that was very nice, because it showed that independent of the specific experimental perturbation Nicoletta induced, the critical point remained the same. The tissue behaved exactly as predicted by these connectivity maps. So essentially what we concluded here is that the central blastoderm fluidization resembles a rigidity phase transition. And the question then we ask is, how is this phase transition spatial temporally controlled within the blastoderm? So the one question we want, before we go in the spatial temporal control, what we wanted to know is, how is connectivity within the tissue being determined? And what would be the function of adhesion in this context here? And this is a very simplified version of the blastoderm tissue, now consisting only of 4 cells. And this is again what Bernard and Eduard were doing. They wanted to put it here in the framework of interfacial tangents determining the size of a contact between cells. And essentially what they proposed is, if these cells are becoming adhesive, then the ratio of interfacial tangent at the cell versus the cell outside interface should change. The contact should grow, as you know, and this would be the contact angle here. And the factor alpha would be then the ratio of interfacial tension at the cell medium versus the cell interface, and alpha would then determine the adhesion of cells and the size of the contact. This goes back to my introduction when I talked about cell-cell contact formation. And now they ask, if you start with a configuration where 4 cells are essentially making contacts, but the center cells are not making a contact yet, then you have a topological structure which is floppy. And now what they did in these simulations is they just increased now the adhesion. They decreased alpha and increased the adhesion. And then essentially you're ending up with a configuration where the center cells are making another contact. And then you go from a floppy topological structure into a rigid topological structure. So this was just important to make a connection between the function of cell adhesion in establishing contacts between cells in close proximity and the emergence of rigid topological structure. So thereby sort of linking adhesion to tissue rigidity. Now you can also see that then if you look at the contact angle, or the ratio of interfacial tension, or the factor alpha, which is inversely related to the adhesion, then you see at the point where the tissue is fluidizing at time point 0, the adhesion between these cells is going down, alpha goes up, but adhesion goes down. And if you see, then you can plot then the factor alpha, adhesion going down in this direction and alpha going up, then you can see that the normalized connectivity goes down. So the connectivity really scales here with adhesion of cells, or the ratio of interfacial tension between cells. And likewise, and this is quite important, the viscosity sharply changes right at a certain cell-cell tension value from below which the cells are losing contact and above which the cells are in contact. And this is the viscosity again following that. That sort of puts together this framework of interfacial tensions determining cell-cell contacts and contact size, and this percolation theory where we are assuming that cells are in contact once they are in proximity to each other. Now the question, and I asked this question a little too early before, is what determines that the tissue is uniformly fluidizing at a certain point? And why is it not just locally fluidizing? And I mentioned before already what leads to the fluidization of the tissue is the ability of the cells, or the fact that these cells are undergoing their last round of synchronous cleavages. And the cleavage of these cells is determined by the cleavage cycle oscillator, which has a core component cycling B, CDK1, and which has a couple of feedback loops which allow the oscillatory nature of this biochemical interaction framework here. And there are two loops which are important. One is a double positive feedback loop with CDC25. And another one is a double negative feedback loop with V1. And this allows to manipulate now this cell cycle, or disrupt the cell cycle oscillator, and thereby disrupt the synchrony of divisions of these cells. So essentially, if you overexpress CHK1, you can inhibit CDC25, and then this oscillator gets dysregulated in different cells. And as a result of that, the cells are not dividing in synchrony anymore. Now, once these cells are not dividing in synchrony anymore, they would lose their contacts not in synchrony anymore, meaning that contact loss within the tissue would become heterogeneous. Now, this was the experiment she did. Nicoletta did then is she overexpressed CHK1. She interfered with the synchrony of divisions within the central blastoderm. In a wild-type case, these cells are all dividing at the same time. But if you now take this subdivision of the tissue, this artificial subdivision in 4 quadrants here, and you look at the spatial heterogeneity of connectivity within these quadrants in wild-type, it would be very, very low. These are the gray points here, because the tissue is essentially losing contacts right all at the same time. Now, if she interferes with the cleavage cycle oscillator, then this is lost. These cells are not losing, they're not undergoing their last round of synchronous cleavages anymore at the same time. And thus you have a very high spatial heterogeneity of connectivity, which relates to the spatial heterogeneity of dividing cells. Essentially, she creates, and this is just a proof of it, she creates a tissue which is not losing contacts uniformly anymore, but in a very heterogeneous manner. Now, what would be expected if you have a tissue and where contacts being lost in a very heterogeneous manner? You would assume that there are clusters which are fluidizing and others which are not fluidizing. The way you can look at it is, again, doing these two-dimensional connectivity maps. In a wild-type embryo, this is a uniform tissue. It loses contact randomly, essentially, in this tissue. And you end up, shortly before fluidization or right when fluidization, you have one large cluster, and then you have many very small clusters. Once you're losing the uniformity of contact loss within the tissue, you don't have this pattern anymore. You have clusters of various sizes, where one is a little bigger than the other ones, but you get all sorts of cluster size distributions here. So what she then did is she wanted to see if the tissue can still fluidize in a sort of uniform way when she's interfering with the cleavage cycle oscillator. In a wild-type, as I've shown before, for fluidization, the tissue is rigid. You have a very large giant cluster. And once the tissue fluidizes, the giant cluster size becomes very small. This is exactly predicted by the theory. Now, in the cases where Nicoletta interfered with the cleavage cycle oscillator, the tissue goes from rigid to floppy, then it goes back to rigid to floppy. It's a very irregular manner. And again, there's no clear giant cluster, the size of the giant cluster is not clearly subdivided between rigid states and floppy states. And she also did then viscosity measurements. Again, in a wild-type case, these are the gray lines here. You see that the tissue fluidizes right at the point when it should fluidize. In cases in which you interfere with the cleavage cycle oscillator, this fluidization happens quite ectopically at different stages, and there's no clear point when the tissue is fluidizing. This essentially just confirms the assumption we had at the beginning that for the tissue to uniformly fluidize, it has to undergo a synchronized last division round or cleavage round, during which contacts are being randomly lost within the tissue. Once you are interfering with this synchronized cleavage round, then the tissue fluidization is very patchy and heterogeneous. Okay, so I'm pretty much at the end. I think we gathered quite a bit of evidence that the tissue indeed is undergoing a rigidity transition. This transition is being triggered by the last round of cleavages, which leads to contact loss. And the tissue then resolidifies after a while again. So this really shares features of a phase transition. So deep cell connectivity determines the rigid topological structures and thus the phase state. And the meta-synchronous cell division spatial temporal control plus the rigidity phase transitions. Okay, I'm pretty much at the end. This is where we are. Nicoletta did most of the experimental work. She's now at the EMBR. Bernard in Eduard's group, he's now at the University of Graz. He did pretty much all the theory. This is an ongoing collaboration we have with Eduard here at IST. That's the lab right now, and I'm pretty much at the end.